do it because it allows you to I'm sorry, it allows you to actually use it for analytical qualitative and quantitative aspect both. Then you have the pharmacopoeial HPTLC, as I told you, the methodology got adopted, and slowly monographs were rolled out from the US pharmacopoeia. Based on this, you have Propitania somnifera, curcumin is there, and many other herbs have been already rolled out into them, and an official standardized methodology of understanding quality control as well as with markers you can establish those methods are there. And finally, in 2021, we are glad to announce that we introduced the first robotic HPTLC in the world, which does this entire process without any human intervention. So you just have to place the samples like typically you do in any other chromatographic system. You have to load the stationary phase, which automatically moves from one module to another module. I will come to the concept of modules and why is this concept of module is there, that also I will highlight. And then it ultimately does the entire analysis by itself, and then the data is also digitally recorded autonomously without any human intervention. And finally, you get the report in your hand. So in the evening, you can go setting up things, and in the morning, you can get all the data in your hand. So you just need to review the data, and that's it. So that is the technological advances which have come up and giving you a lot of potential to work into it. Now, when I was talking about TLC and HPTLC, this is the vast difference. Typically, you have something which is done by spotting, and left-hand side is the concept of HPTLC, which believes in resolution of compounds, which believes in reproducible result, which believes in to give you high throughput so that when you want to quantify also, your results are absolutely perfect. And that is established with the help of very, very much standardization, highly standardized procedures with the help of machinery and instruments. And that is how it goes ahead. So when I talk about HPTLC and compare it with TLC, this is a huge difference. So there was the research which was done, and the size of the plate was seen that in a small resolution of plate also. I talked about stationary phase, you remember, and this is the stationary phase where I am separating components. This is a fluorescent plate, so the background is absolutely dark, and we illuminate it with 366 nanometers of light, where the spots are being observed with fluorescence. And you have a standard, that is, you can see on the 9-number track in case of HPTLC, the right hand side plate. And on the left-hand side, typically, a TLC plate uses a bigger size, because of which the development time required is more. You see a lot of diffusion, and the resolution is not of that great importance. So here, in smaller size, you achieve the same, keeping the resolution at par and highly defined spots, which you can see. So this internally, what it does is less solvent, less time, sharper bands, more sample. So you see, it has involved itself, giving you all these advantages. You know, all these things indirectly materialize to your time, and time is money in today's world. So every time you say that how do you justify the cost, it is this answer that it is giving you a lot more output in a very short, in a very short span of time, and that is how it works out. Am I audible? Hello?
Yes, sir. Yes, sir. Pretty good.
Yes, sir, you are audible, sir.
Yeah, am I audible? Hello?
Yes, sir. Audible, sir.
Yeah, I'm extremely sorry. I think my laptop connected with an alternative device, because of which the voice started off there. So did you miss anything, or this slide was explained by me?
Yes, sir. Yes, sir. It is okay.
Okay, thank you. Okay, So getting back, as I told you, this accounts to 4 more samples. Now, when I am in a position of a researcher, my thing is that I have many of samples. I need to analyze them in a short span of time and get the result out of it. And HPTLC solves this, problem of it. You can take lots of sample, analyze it in a very short time and get the results, because one typically this size of plate takes to around max to max 1.5 to 2 hours, and in 2 hours you are getting as many as 15 to 20 samples output. So that is the power of this technique, without bothering that anything will get damaged. And this is, I'm talking about if you are doing just one experiment. If you are doing simultaneously 2 experiments, it goes back to back without occupying the instrument for one person. Immediately once step is done, the second person can do that same step without intervening into the first person's analysis. So it becomes that in the same unit time, say around 2 hours, 2.5 hours, you can do as many as 75 samples and get an output from them. So that really speeds up things, because at an analytical, when we used to do our PhD and we were struggling for something faster, which could analyze many samples, many of the times we are worried with 2 essential parts, like the costing part and the time part. And because of this, we refrain to take more samples. Now, if you see the statistical books which are there, they always recommend you to have at least a group of 6 samples to give you a proper conclusion. So 6 is the minimum value of each sample which you should analyze. Let it be a plant based, let it be a pharmaceutical concept, where it be a cosmetics, but you should take at least 6 different samples and analyze them, and then only conclude your research work. And with most of the analytical people, this becomes really challenging, because they are not able to perform that in the same unit time, either because of the cost or because of the time they miss out doing that. But HPTLC solves this, because if you are at a screening step or you are in the quantification step, use this and utilize it in so fantastic way that you get the output in very short time. So analyzing a lot of samples is never a problem. The economics is cheap, and also the time consumption is less. So both advantages are there. So that becomes one of the better choices for the analytical world. Now, as we move further, we have the data output. That is, now I'll explain you a little bit about the instrumentation. Now, this is how the instrument looks. We have a modular based equipment that is there. When I talk about HPTLC, it is not that one entire instrumentation is there and that becomes your HPTLC. It is a serial of modules which do stepwise analysis and give you a final output. Like here you have the first step, that is the applicator, which does the primary job of applying the sample onto the plate. Now, a lot of science is also involving into it. I will come back when I talk about more in the application. Now, typically when we do an application like spot, what we do is we'll spot it. We'll either allow it to air cool, or maybe in sometimes blow also air over it, so that it quickly dries up, and then I can, if I have to load a more amount of sample onto it, I have to give multiple spots over there, and this takes a lot of time. So in our applicators, we have a standardized technology of spray-on application, where the sample is actually sprayed onto the plate. Now, the spraying gives you one, uniform distribution of the particles, and second, it dries the spot very fast. This gives you another advantage, is more loading quantity onto the plate. So at the same time, 3 advantages are available to you when you go with these applicators. There is, again, from the basic one to the automated ones, which do the rinsing step and do the application by itself. I'll show you when we go into the demonstration part. Then we have another module, which is the developing device. Now, this device also has multiple things which it does. So first of all, after the application, it will first condition your plate surface with the set humidity. Now, humidity is one of the important aspects in HPTLC, which is one of the parameters why variation is observed. So what happens is the surface, it absorbs moisture, and because of which, when the band is being separated, it gets diffused because of this water presence onto the surface. So that is being controlled with the help of this ADC2, it conditions this plate surface with the blast of an air, which is conditioned. This air has 33% moisture level. So the moment it touches the surface, it removes the excess moisture, absorbs into it, and leaves. And again, there is a chamber inside where it goes, deposits the excess amount of moisture over there, and returns back onto the plate with again condition 33%. Now, this is achieved when we circulate the air over a saturated salt solution, and one end is the plate and one end is the salt solution. So the air is coming, picking up the moisture, going into the salt solution, depositing the excess moisture, and coming back. This salt solution generally used is magnesium chloride hexahydrate, which we prepare a saturated salt solution. And in this ADC2, we have a device known as the humidity control, which has this salt solution inside. So secondly, first, when you finish the application, it dries the surface, removes the excess moisture, and then it does the plate development autonomously. The solvent pours into the chamber, the plate goes inside, the development happens, the separation happens, and the moment the solvent reaches a certain level onto the stationary phase, it identifies with the help of its sensors and tells the system that now the plate is being developed. You need to remove the plate, and it removes the plate from the chamber. And after that, it does this final job of drying the plate. So it is also taking care of drying of your plate, where you can divert it to a fume hood, and the excess solvent which is there is quickly dried off from the plate surface. That is how it does all these 4 things autonomously without any human intervention once the command is given. Whereas in the same manual thing, we do it more manually. We take the plate, we dip it, we ensure that we dip it in the proper way, because a slight tilt would result in a tilted solvent front. My molecules, instead of being straight, they will become tilted, and because of which I will not get my results properly. So that I have to be careful. I have to condition this plate by keeping in a desiccator with saturated salt solution, expose the plate for 45 minutes, which is done in this case in 10 to 20 minutes, and then the plate is ready. But when you do it manually, it takes 45 minutes. Then remove the plate, and then you develop with the mobile phase, and you have to monitor by yourself. And after you remove it, you have to dry it yourself and take it to the next module, which is the visualizer. Now, visualizer is the place where the documentation happens. The image which you are seeing is under 366 nanometers, where the background of the plate is dark and the bands are fluorescent. Now you will say there is another wavelength which is 254. I'll show it to you as we move into the instrumentation, where the plate background is bright and the spots appear to be dark. That is known as the quenching. So the plate has an minder in it. There is a UV active compound which is added into this plate surface. So it fluoresces when exposed to the 254 nanometers of wavelength, and the quenching that is there, that is being absorbed, that is the actual band. And with these 2 techniques, we are able, and even the white light, after white light, you see the bands visibly. That is also being documented with the visualizer. And after that is the crucial part, that is a scanner. Now the scanner is more tuned in to do quantification accurately. It has a wavelength from 190 nanometers to 900 nanometers. So it has the entire wavelength range, right from UV to visible, and a little bit of IR part that is there, but a negligible one. But it covers the entire range to give you a proper wavelengths which are required. For example, caffeine, that has a wavelength lambda max of 276. So when you scan it at 276, the background noise gets totally submerged, and only the molecule of my interest is being highlighted over there. And that is how you increase the sensitivity of your analysis and do the quantification rightly, more accurately. Also, it has got features to do spectrum comparison, that is a spectra pattern obtained at different wavelengths of a particular, like this spot, for example. If I have any spot and I just want to know the absorption maxima of it, I can set my system and tell me whether what is the absorption maxima. It will scan from 190 to 900, it will give me a pattern, and I will just compare it with my other component and come to a conclusion, like whether it is matching with my sample or not. So those all things are taken care by the scanner. Then you have the TLC MS interface, which is actually lifting the molecule from the plate, and it is a type of hyphenation. I told you, you can inject the molecule directly into an MS or collect it in a vial. Now the advantage over here is you have least contamination. That is very important, because when you are handling an instrument of mass spectrometry, which is a very sensitive detector, and you inject anything into it, it will give you something, some noise will come in. So if you are contaminating and giving a lot of contamination into it, it is bound to give you a lot of peaks, and you will never understand what is there. So this TLC MS interface minimizes that complexity, and only the selected molecule is being injected with very, very low background noises which are there, and selective molecules can be identified with the TLC MS interface. Now, people may think this is very simple, like you just take a plate and send it into a mask, but it is not that. The mask is a sophisticated instrument. You have to first tune the mask to see whether it is responding to your requirement. For example, if I'm searching for an alkaloid, I first need to ensure that my mask is tuned to give me a response of alkaloid. If it is not, then even if I inject an alkaloid into a mass system, it may not show me the proper parameters, because there are many things into a mass like cone voltage. We are not there to discuss more, but the criticality in mass is much more higher. So whatever you're sending into the mass has to be at purest at form, and that is why this TLC MS interface helps you out with that. And after that, you come with the last component, that is derivitization. Now, this is an interesting concept. Many components are not visibly seen or are at low UV, say 200 nanometers. Now, what is the concept of low UV? It is like my components I am separating on the plate, but I'm not able to see either visibly or in the UV also it is not being detected. So how do you understand whether it is there or not? So I do a simple step. I take a derivatizing reagent, which is normally a chemical mixture. I spray it, either spray it or I dip into that. These are the two popular techniques which are being used. And then you just dry the plate and heat the plate in a plate heater. The plate heater will actually escalate the process of reaction, and slowly you can see the spots. The last example which you are seeing above the derivatizer is that image which has been derivatized, and what it does it now your molecules become visible. So not only I can identify such unknown compounds, but which are not visible or seen, but I can also get an information out of that. For example, if I'm searching for an alkaloid, I can specifically use an class specific reagent for that particular alkaloid, say drag and drop reagent, and it will highlight me that particular spot as red, orange, or and it will tell me whether there is an alkaloid presence. So preliminary, when I have 100 things to screen and I have no understanding of what it is, I can explore these derivatizing reagents and find out whether it is a Kaumarin bitter principle and things like that. So this can be a really game changer for understanding of what I'm searching for. This derivatization has also increases your sensitivity, and it can reduce your LOD and LOQ values, which are critical for quantification. So after moving to the derivatizer, we move into the next part, that is the software, which not only controls this equipment, it stores the data for you in a nice printable format, and you can use it anytime when you want to do a presentation or submit the analysis to a report. You can do that. So this is how the HPTLC output is there. The quantification. The quantification is an important aspect of HPTLC, and many people think that how much low I can go for a detection, what will be my LOD, LOQ values, because in other formats of chromatography, we get a lot more higher sensitive. Sensitivity is first of all also a concept. If your instrument is more sensitive, you are more bound to get errors. That the more number of decimal points, the more error chances increases, because you may, what you are accurately analyzing may be plus or minus more in case of a very sensitive equipment. But when I say my detector response is within a small range and I have very less range of deviation, so that gives me more accuracy towards my analysis. And here exactly what we talk about is the sensitivity. Now we have mentioned over here as nanogram and picogram. So that is under UV absorbance. So the example which I showed over you there was a fluorescence plate. The earlier example which you see on the left hand side topmost, that is a fluorescent plate, and the UV absorbance, which is the greenish background because of that activated zinc which is there, which induces fluorescence to the plate surface. That in that format you can go at nanogram levels. But when I talk about, people always correlate this to ppm and ppb. In HPTLC there is no concept of ppm and ppb. However, your sample preparation will be in ppm and ppb, but once it lands onto the plate, it is an absolute concentration. Why? Because you are doing a spray application, the solvent is evaporating and the molecule itself is depositing onto the plate. So it becomes a direct weight that is being deposited onto the plate. And that is why we talk about nanograms and picogram. So when I talk about ppm and ppb levels, so my sample preparation, I normally start with mg level. Okay. So mg level, I weigh something, and from that I need to detect it at ppm level. So from mg, how much low I should go? I should go from mg to mu g, and from mu g to nanogram. So that becomes my ppm level. So if I have anything prepared in mg of my standard or sample, I can easily go for my ppm studies onto HPTLC, because nanogram is easily being detected. Secondly, if anything is in picogram, so then it is like you are going 10⁹. So as I told you, it will be ppb levels also you can investigate. So normally, if I'm starting at mg, this is at ppm and ppb level. But if I'm starting at gram, I can go at parts per billion and trillion also. So technically, to investigate into it, HPTLC is really not difficult. You easily able to detect the compounds which are at ppm and ppb level on HPTLC. There is absolutely no problem. Secondly, we have the advantage of loading the compound more. Now, in many other formats of chromatography, you have to limit the amount of loading because of the damage associated to the stationary phase. But in HPTLC, because our plates are disposable, because you understand what is happening onto the plate, your excess quantity also, if you load, it will be showing you where it is, whether it has stayed at the bottom, whether it is preventing any separation. All these things can be identified. And to most of our cases, we have found out that loading even large quantities for impurity profiling does not hamper our HPTLC stationary phase performance. It separates out. We can easily identify that impurity and tell the percentage of that impurity at 0.001% also, or with our HPTLC system. So that way, sensitivity is never a problem of HPTLC. You can easily use for your all kinds of analysis that are requirement. Other things I have mentioned, more things like accuracy, precision. Of course, these are instrument based. When you use the instrument, you get these values, and that is why all the methodologies which you have seen are developed with instrumentation to give you the highest grade of accuracy and good quality results. Then we have MS hyphenation also, which can be done for identification and verification. And finally, regulatory compliant. HPTLC system is universal in nature. There is no different models for different country. Like in India, we have a separate system. In abroad, we have a separate system. Everywhere it is supplied with the same standard specification. And that is why what you produce over here, it is easily accepted in other countries, because you have done the same thing which they would have done in their equipment. There is no change of other things. Whereas in any other chromatographic system, you have to match the result, and specifically that instrument has to be there to approve your results. Otherwise, your results will not be approved. So that problem is also not there because of being the universal nature. So these are some of the question answers which normally come up against quantification, and I have discussed them, the most important ones. Others are self-explanatory, I can talk about. The concept of fingerprint. This is interesting because this gives you a lot of information. Now, HPTLC, as you know, is firstly a visible based. So whenever you're doing an experiment, the first output which will be coming is by you. You will be the first detector. And when it is done by human, the error is I would not say it is minimized, but it is the accuracy, the understanding is more. And AI will take time to adopt, but an human will tell you whether this sample is passing or failing immediately by observing it. So the first detector is you. That is the basic advantage. Whatever happens of my sample, 100% accountability of that sample is in front of me, because I know what amount of sample is at the bottom, what amount of sample is at the top, and where is my marker coming in, where are other components which are coming in. So I am in total control of understanding how my sample distribution is there onto this surface, and that is why I am 100% accounting that my total sample is I am accounting. If for any case my certain component is at the bottom and only few components are coming out, I immediately come to understanding, and without proceeding for the quantification, I will do is I will change my mobile phase. Repeat it. It's a function of polarity, where you have compositions, mixtures of different polar solvents together. And if at a certain polarity you're not able to elute something, you change the polarity, you develop it so that the molecules which are at the bottom match to that polarity and come off and get separated. So that is quite possible. And that is why there comes the concept of fingerprint in HPTLC. A fingerprint is something which is a unique pattern of that particular compound in that mobile phase to give you separated compounds. This is very interesting. This gives you first glance impression of the sample, whether it is passing or failing, whether my marker is coming out, whether my extract is proper or not, whether my formulation is proper, whether any impurity is being generated. So many things you can find out at a glance. And apart from it, you can always take a document image and go for the scanning or derivitization for further confirmation, even an hyphenation. So this becomes 100% of your sample in front of you, and that is where you get the maximum advantage of this type of chromatography. One example, I've definitely chosen the herbal example over here because the herbal matrices are much more complex. So normal, pharmaceuticals are 1 or 2 or 4 or 5 components at the max, so they can be easily resolved. So that is why the most complex one can be resolved with this. And this is an example of different types of black pepper which are available in the market. We compare the profile and come to an understanding how their profiles are different, whether my sample which I'm getting is homogeneous, the quality I'm able to standardize. Up till now, what we used to do is only see the marker, and I would only see the external appearance, and I would be happy that I am getting Piper. But imagine in 100 kgs of sample, if I have 10% adulteration, it will give a lot of profit to the vendor. But when I'm preparing certain medicine or something out of it, similarly, when I'm having a batch that is 90% A and 10% is not failing, and I can establish this, that yes, this 10% intensity less I am getting, that is being proved with the scanner, and quantificationally I can say that this sample is not at 99% purity, but it is 90%. Then piperine will come, obviously, because pepper is there, but the pepper quantity is less. So that is why, and nowadays, spiking is an easy feature. You just spike in and you get the same value, and easily it can be fooled. But this fingerprint can never be fooled. It gives you a clarity of picture, and you can immediately come to an understanding whether it is matching or it is totally different. Why is that difference is there? That also we can find out by doing a national survey, different parts from the different vendors, different samples, and that you can compile and see how much is the variation. Time points are there when the sample is being collected and all those things. So if properly done, we can actually standardize this. The monographs which you are already there in USP, these studies have been done, and you get a very standardized monographs of this. And this is in-depth study which you do and get. And for doing depth study, you require a lot of samples. In any other chromatography, that is not possible. But in HPTLC, it is possible. You can do many samples without getting into controversies, easily done at anywhere, because the structure is universal, the instrumentation is universal, the plates are universal, everything is universal. So now, as we move ahead, again, there are some basic concepts. Now you must have realized how HPTLC is totally different from TLC. I'll not go into this again. These are again some more advantages. Now the data output that I would like to highlight, you get the more number of data points. Imagine you are plotting a graph, and from that graph you are given 3 points, and from that 3 points you are you are preparing one graph. When you prepare the graph, if you have more number of points, your graph is much more accurate. The lesser number of points are there, the graph is less accurate. So when you have a molecule and you get all this data like UV, vis, fluorescence, spectra, derivatization, MS, and bioautography, that is effect directed analysis, that is known as, if you have so many of data outputs of it, imagine the accuracy of predicting what that molecule can be, or at least understanding what that molecule can be increases at last. The knowledge of that molecule increases at large, and that is why you are in a better way to understand things. So this precisely gives you a lot of advantage, as I was talking about. For the practice of HPTLC, we have seen that diagram where I show you application, different modules are there. Now the approach is also the same way, standardized approach. Excuse me. Software activation, application on plate. Then you have chromatographic development, image documentation, densitometric scanning. Excuse me. Then after scanning, it is non-destructive. So at this point of time, your molecule is on plate and it is available for doing other kinds of study. It has not got any degradation or anything. Simply that molecule from the soluble form is placed onto the plate, and now it is present on the plate. After you finish the scanning, you can do multiple things that are destructive in nature, like the molecule will change after that, and either you go for an MS or IR. Now you will say how IR and NMR can be hyphenated. The same way I told you the TLC MS, if I don't connect it to the mass, I can collect it in a vial, dry the amount, and now I have that molecule with me, which I can use for IR and NMR. Of course, in this, when I talk about isolation of compound, you have to know the label claim of the compound. See, if it is in 0.1 and 0.2%, for that you have to load more quantity onto the plate, or if it does not support, you have to run multiple plates 3 or 4 times, what is known as micro preparative work or preparative work. And you have to concentrate that isolate, because here you are in a better control, because each and every time you are getting the same separation pattern and you are able to identify the same substance, collect it in the vial, try it. Now you get a very concentrated format and you can do further studies which are destructive, as I told you. You can go for the derivatization also. Bioactivity, I will also explain you how bioactivity is being done with the approach of effect directed analysis. So the effect which I am targeting, based on that I do some analysis, and from that I get an efficacy output. So no need to get ethical committee meetings and all those things. On the plate you can screen several samples, several isolates on HPTLC and come to a conclusion. This is my choice of interest and I can go ahead with that. So that is that simple. And then you have you can perform again imaging and documentation for derivatization and bioactivity type of study. So it is very approachable. There are many other reasons why it becomes ultimately and choice. And there are, modular study helps in many people doing simultaneously. We have 1 or 2 sets of equipment at our facility, and we use around 14 people simultaneously. So our work does not get hampered, because one person is doing application, second person in development, third in EA. So per unit time, we have a lot of output which is coming in. That is the beauty. One equipment with many person use in a single time gives you a lot of advantage for the output part. So that is one of the advantages, solvents and everything. When you are doing research also, the solvents are costly, and that is why one bottle over here lasts you for many experiments, because you just consume 30 ml per year. And of course, being an HPTLC specialist, our expertise is always available to you any point of time. You can write to us, get a touch to us, and that is very important, because when you are working with something and you are stuck up, and help can be something which can help you out, come out from that problem and solve it easily. So that is why our expertise is always available. That is what Anchrome is all about. So those earlier slide was talking about that. And now when we move ahead, I have explained you about different chapters. These are the gist of entire chapters what they talk about, and USP chapters which are there. It is also there in the WHO monograph, Chinese pharmacopoeia, European pharmacopoeia, and in Indian pharmacopoeia also it has got introduced. So this is about regulatory compliances. So absolutely no issue using this equipment. These are some of the essentials which are required: fixed sample application, chamber size, humidity control, which I already talked about. Resemblance, we'll go through this as and when we move further. Important part over here is the IQ and OQ. HPTLC systems are under compliance of IQ and OQ. So IQ is installation qualification, OQ is operational qualification, and there is also a PQ, which is performance qualification. All the instruments are qualified, and it is electronically tested by the software and manually tested by the user to give a proper IQ, OQ, and PQ document. So it is compliant with all these tests. We also have a system suitability which has been launched. There is a dedicated paper of a system suitability, and variations can be checked with system suitability approach. I refer you to read this paper, which is freely available on universal mixture, UHM. It is known as UHM mixture. You can type it for TLC, thin layer chromatography, or HPTLC, high performance thin layer chromatography, and you can get a lot of understanding of this. So system suitability ready mix, which is available, and you can use it for your analysis. So that also has been simplified if you want to perform the SST, that is system suitability in HPTLC. This is the standard approach of 1064. As I told you, everything is defined right from how the plate should look. Of course, when you are doing an R&D, you need not to follow these. Only when you are going for the publication part, at that time, you need to input all these details. Spots, bands, band length, everything has been standardized, and thereby you know that each and everything gives the variation in HPTLC, and that is why this standardization is mandatory when you are doing HPTLC document, and each and every step. Max to max is that you can reduce the band length and you can accommodate more samples, and the length of mobile phase may be developed, change to 90 depending upon the resolution of compounds which you require. But other things like saturation and all band lengths, we recommend you to follow strictly the HPTLC USP guidelines. You can read through this, as it's freely available online for more. And as I was talking about, this is the next generation of our equipment. But before we move into this, let us go further to understand, and at the end of the session we'll move in through the practical part. This is how the universal approach of sample preparation is there for botanicals. Like, you take your sample, if it is a powder, you prepare 100 mg in 1 ml with methanol, and if it is there that 10 mg, if it is an extract, like already it has been extracted and sent to you, and analysis has to be done, then you prepare 10 mg per ml. For other pharmaceutical aspect, it depends. Chiefly we start with 1 mg per ml, that is a 1000 ppm standard, and from that we dilute it to 0.1, 0.2 ppm, sorry, 0.1 mg per ml and 0.01 mg per ml, and see the detector response, and based on that we perform the experiments. But for herbals, this is the general approach which we do. The performance of sample preparation is also very simple. In centrifugation, you can get thoroughly clean up, because HPTLC can tolerate more matrix. You don't have to worry that the sample to be very much high purified, because ultimately you have the polarity in your hand in the next step to elute what you are interested in and retain what is you're not interested in. For example, if I have a mobile phase for gallic acid, it will be more polar in nature. So all the non-polar things will migrate on the top, and what I will have it on the between. Similarly, for flavonoids, I'll have a mid-polar mobile phase. So only the mid-polar components will be coming in the middle. So like that, you have this strategy, and that is why this simple sample preparation is enough for our HPTLC analysis. Then this talks about modules. Now, this is the very basic equipment for applicator, where you have a semi-automated one. Although the same task is being performed, you can precisely and accurately regulate how much volume of sample or standard you're loading onto the plate. Up till now, it was not possible because in capillary, we used to get variation. Sometimes a person used to say, I spotted 2 times, but 2 times is how much? How much volume you have? There could be no answer to that. But now you have an answer to that with the fixed volume syringes, which has a gradation, and you can load fixed amount of samples onto the plate. And later stage, when you are going for quantification, this helps you out. The dispensing is fast, the sample application is fast, and the next step preceding it becomes very fast because of all these advantages. These types of application normally are done with these applicator, single applicator with all these types of approaches. Even micropreparative is supported, so you don't have to buy anything new for doing micropreparative. Our applicators are designed to do basic HPTLC to micropreparative work also. And a normal approach, the quantitative standard sample beside them, and then you compare and get the results. In situ cleanup, like you have an oil sample, directly you dilute it with N-hexane ethyl acetate, apply it onto the plate, and then you develop it with petroleum ether. All the fat goes on the top, cut the plate at the bottom somewhere around here, and then tilt it at 180 degrees and then develop. So all the mid-polar components of that medicated oil are preserved, and now you have the second mobile phase, which is mid-polar, and there you get the separation. And after that, you have the micropreparative isolation kind of work, where you apply a single band, a large band, you develop them, and you can now go for the isolation part, as I told you. And we have an interesting feature of superimpose, where you take one spot, apply the spot with a known either standard, or it may be an internal standard or an external standard, and see whether the sensitivity is there. Sometimes we say that it is not being detected. So we have a question whether my sample, whether my chromatographic system is able to separate this component. For that, what we do is we intentionally spike a certain compound onto the plate and we separate it. Now, this is not a recovery. It is recovery, but it is on plate recovery. It is not sample recovery. If you say wet chemistry sample recovery study, that is in sample you spike, then you perform maybe successive solvent extraction or the same method of extraction, and then you apply. That becomes the wet chemistry based sample recovery. But when I'm talking about this on-spot application, this is on-plate recovery, which we are checking. So if the standard and sample are present in that, whether they are getting separated onto the plate, that is being checked with this particular feature, quick and fast. And also in certain cases, it tells you whether detection is being missed or not. If it is in very small amount, and suppose you have a slight suspect that it may be present, so what we do is we apply a known concentration of standard into it. That known concentration should give a known area, which is compared to that pure standard area. And from that, if there is a difference from the area received from the sample and then the standard, we come to a conclusion that yes, there is if there is an increment, definitely the standard is present, but at this much amount. And if it is not, if it is low, then definitely the recovery is break apart, and there is some problem with the chromatographic system or the sample extraction, which needs to be tuned in. So both the ways it works, and with our applicators, you can do this smoothly. Then you have the development part. In the development part, the plate once has been applied goes for the development. We place the plate in a twin trough, we develop it, and after the development, what happens? This separation of the compounds happen. After the separation happens, we see those bands, and from that we get to a conformation. Now this is happening again with different instruments. The manual one, the left hand side is the ADC2, left hand side bottom, which has that small glass chamber, if you can see, which is for the humidity control. Then we also have an automated multiple development device, which is a kind of reflection to gradient aspect. So you have mixed polarity components. How do you separate them? You change the mobile phase for a different length. So if this is my plate and I need to separate this component, so for the first mobile phase, I'll develop it this much distance. Second mobile phase, I'll increase the distance, and I will change the mobile phase according to the polarity, and I will get these components separated, that is multiple development and also the gradient type of development in HPTLC. We can do this with our instrument, which is able to separate as many as 44 compounds, phytochemicals from a certain plants with 25 gradient steps that have been tried, but it supports 1,000 gradient steps by the instrument. Then coming back, all the instruments are compatible with any kinds of solvent systems, any kinds of stationary phase. So gives you a lot of flexibility. I have a glass plate, you can use that. I have aluminum plate, you can use that. I have a plastic plate, I can use that. I have a silica plate, I can use that. I can have reverse phase plate, I can use that. I have a preparative plate, I can use that. So n number of plate dimensions which you have, all those can be used on our systems with any combination of mobile phase, non-polar, polar, mid-polar, any combinations of mobile phase. So you get a lot of probabilities and doing criteria. Okay. One of the things over here is when I start with, I do have confusion in my mind how to start with which mobile phase. So for that also, TLC by itself has a lot of information available on the net, as well as there were several books like Egon Stahl book is there, C.V. Clark book is there. Then you have the P.D. Sethi, Dr. P.D. Sethi's book on pharmaceutical analysis is there. So they have a large range of mobile phase which can be compatible and you can use them also. Joseph and Sharma, Thin Layer Chromatography by Joseph and Sharma. These books are very essential and you can get a lot of information for your research work. So starting point is never a problem in HPTLC. Then this is the module which I was talking about, the HPTLC. And some people say that if my molecule is stable, unstable within the mobile phase, it is degrading, or whether it is degrading, how do I find out? So normally this is based on multiple development only, but what it utilizes is different dimension development. So what happens is that same molecule, I develop it in one direction, then I rotate the plate by 90 degrees and I develop it in other direction. When I do that, the components, if are related, it will get separated onto a single line. If they are degraded by the mobile phase, they will degrade it. They will show you outside the straight line. As you can see, it is written as unstable. Now this happens either the molecule is unstable on the plate or the mobile phase is degrading the molecule. In the first case where you see the stability, the molecule is already being degraded and it is being moving after the mobile phase has been developed, but it can resolve your degraded product. So both the types you can get different information with a single approach, and this is how you see them. Now, as I was talking you about the gradient development, this is what about different steps. You can see with each step, I'm changing my mobile phase and the distance. The same molecule is getting separated as per the mobile phase. And so you can do at max 25 is the practical part. But as I told you, the instrument suppose 1,000 steps which you can give. And this is one example where phospholipids. Now, phospholipids are generally our skin lipids which are there, or some these phospholipids are also being used for drug delivery systems in your micro. I'm not able to exactly find out that word, but I think it is named as microcapsulations and things like that. So here you can also analyze them and find out. So that is one example. And now, as I was talking about the images, I was talking about the 3 kinds of images which are generated. So by default, the plate, if it is UV active, either it will show you the bottom one, that is the fluorescence, or the just above the bottom, the greenish fluorescence that is there, that is, it will show you the quenching effect, that is the dark bands which are there. So if it does not show you response in both these two, you can go for the derivatization process. And if you have an access to a scanner, then in UV also you can scan it at 200 and find out, because our scanners don't get any interference from any solvent. We can go really low and scan it at 200 nanometers and get the information. So hidden molecules are easily studied with that. So this is the instrumentation which documents the image. The image capturing device is also specially being made, because one of the challenges is taking photograph in low light and without any noise. So these images are HDR images with a calibrated area. So with a click, you can find out the RF value. You can increase the sensitivity of the image with a clean plate function. And there are many functions which are there which can be utilized with this derivatizer. But the data which is being generated is very large also with the visualizer, and you can do many things with that. So these are some of the things which it does: image correction so that the light distribution is uniform. This comparison viewer is achieved with this visualizer. So multiple samples have been done at different points of time. You need not to run each and every time the same sample. What you need is this software. It will digitally traceable. All the image components are traceable. From which plate have you taken this image? Is there? It is not a Photoshop image, and it will tell you, okay, yes, this correlation is there over the time period, January, February, March, different time points, how much variation is there. Sometimes as a researcher, you may not find the sample exactly on the same month. You are delayed for a few months, and then it comes up, but then you are worried, my standard and everything, how it will be there. But those worry are not there. Simply apply it and use this feature to compare it and find out how it is correlating. You can do this with image, you can do this with the chromatogram, and you can do with the spectrum. I will also talk about more about the screening. Unknown sample screening. This is the way how even the autobiography studies are done. So the same approach, instead of using a chemical derivatizing reagent, what we use is a specific either enzymatic solution or a bacterial solution, or I can use a chemical solution such as DPPH, which is used for antioxidant activity detection. So if my extract has any antioxidant potential, I can find out immediately over here. And also, if I want to be more specific, which component out of it has an antioxidant activity, I can pinpoint with a single experiment. Similarly, I can find out antimicrobial activity. There are many studies which have been reported for different enzymatic assays also, like anti-diabetic, anti-Alzheimer's. These have been reported and papers are available. You can easily perform them if you have a good microbiology lab. This can be done and you can find out lots of information out of it. Then this is a scanner, and as I told you, it is the place where we get the chromatograms. Now, all of you must be knowing about chromatograms. Chromatographic impression is the digitally or electronic signal of what you see at a quantitative level. So what it does is it sees the intensity of the band, and according to the intensity, it gives you this peak. And this peak intensity is then correlated to the concentration to give you a correlation and linearity and things like that. You can do a limit test, internal standard, related substances. All this can be studied with the software itself directly with this equipment. And the bottom half, you can see this is an example of the spectra. I do have an example of that. This is how it works. This is the way the light of selective wavelength is directed onto the plate and the response is being noted. This is the spectra. Here you see the spectra of different components, and each spectra is different because these are of different components. If they overlay, then I will be into an impression that it is the now mind it, I am talking about a single component. Like here, if you see, there are several peaks, but when I'm talking about the spectra, I'm just considering one peak out of this, and I'm comparing that peak with my standard and sample. I'm not comparing many peaks at a time. That can be also done, but right now when I'm explaining you about the spectra is per peak based, that is per component that has gone separated. So this is an example of how the overlay can help you out segregate and understand whether it is matching with your sample. It can be used at identity and purity. Purity is a function where you can find out if it is co-eluting at different points. Multi-wavelength scan is there. So this tells you which component is responding at what wavelengths. And this is crucial for fingerprint type of analysis. And also certain impurities where maybe you can take a wavelength of 200 and scan the pharmaceutical product, and you scan at 254, 260, then you scan at 300 and see the pattern, which components are shown earlier, which are hiding. So what components are present, absent, 100% accountability of the sample becomes over there. Now this is an example at through the software this calculation is done, and this is how the chromatograms are being generated. This is the left hand side, and the best part over here is when we select a specific wavelength, the background noise is highly reduced. Now coming up to the next stage, that is the derivatization. Here with the derivatization we have the plate heater. This is the spraying device. I have a video explaining how this procedure happens. It uses very small reagent, and you just spray the component onto the plate, and we are able to see not many components from it. So this is a derivatizer, and with this we can analyze many components and see what are hidden objects. I have already explained you about what the derivatizer is, and this is an example of that. I told you we do a lot of R&D just to understand what type of a molecule, whether alkaloid is there or anthracene, bitter principle. So we have a set of 15 mobile phases designed with 15 different derivatizing reagent to give an identity what components are present, sugars, saponins, steroids, if anything is there, will be detected, and this is how it is being done. Then we have the TLC MS interface. This is how it works. There is a pump and the mass spectrometer. So the head locks onto the position. The solvent from the pump comes in, it dissolves the molecule, and directly that analyte, after getting filtered, there is a filtration frit inside, goes into the mass, and you get the spectra out of it. So very simple, very fast and rapid. Within a minute of 3 minutes, your one sample goes into the mass. That is one spot from the plate or one band, and you can analyze them back to back, many samples, and come to a conclusion. This is a defined chapter of USP, which gives you understanding of how in dietary supplements TLC MS interface is being used effectively. And this is an example of synthetic tadalafil being added into an Ayurvedic sample, and that is being tested. There are some joint ventures. So our TLC MS interface is universal. It is compatible with Shimadzu MS. It is compatible with Waters. It is compatible with Thermo. So there is no limitation in which MS you can hyphenate it. Single quadrupole, multiple quadrupole. But as I told you, you need to tune the instrument first to get the response, and then you need to inject the sample. Otherwise, it does not detect the sample. So you have to tune that. And then you have the effect directed analysis. That is, this is one other additional product, and generally it is used for toxicity testing. Any kind of toxic substances there, that is based on, effect directed analysis, where the molecule is being applied and developed. Now here the approach is different. The bacteria itself has a fluorescence activity, but that is known as bioluminescence, and when it interacts with the toxic substance, it dies. So this bioluminescence is very very faint to detect. It is like at night when we see some radium glowing. It is similar to that. And what you do is you take this image with a highly specialized device, the earlier which you saw, the luminizer, to capture such low bioluminescence and understand whether the components of the sample are toxic or non-toxic. It can be adopted for many uses like toxicity of water, material, foods, beverages, electricity, and so many things. Then this is another example where effect directed analysis is hyphenated, and here you have this culture of bacteria. And if you see after this third unit on the top, the fourth unit is actually an immersion device. The plate is being loaded and it moves into an immersion device where the bacterial culture is there. It removes, you have to give some incubation time, and then just observe the zone of inhibition, or if it has got bioluminescence activity, you have to observe it under that bioluminizer. And once you have identified, what you can do is you can prepare a similar plate, and that same RF value can be sent to a mass to identify that compound. So very simple to use and a lot of things. Now we'll go into some applications, and then we'll move on to the final part of seeing few videos to understand this procedure. So first is I'll just talk about few minutes with method development and validation. In HPTLC, in most of the colleges, students are required to publish papers with validation and all. So HPTLC supports all kind of validation requirements that are there. You can do all the parameters that is there. Validation time is less as compared to HPTLC because the output is faster. So in a short span of time, you are able to get more output. So typically we finish a validation process in 3 to 5 days at our laboratories. And once the method is ready, this process happens in 3 days and very rapid and fast. In a day, 6 to 9 plates are easily being done, and you generate a lot of data with HPTLC. So method development is also very cost effective. Plates, are cheaper. So a lot of things can be done. And with this approach, a lot of analysis can be done in a month's target. Validation wise, all the parameters are covered: specificity, precision, accuracy, linearity, limit of detection, quantification, ruggedness. LOD, LOQ, there are different approaches. You have the theoretical aspect, which is done through an Excel sheet. It predicts the value, and you have a practical aspect where you actually spot and find out the actual LOD and LOQ values. And ruggedness also, it is there because you are doing the experiment throughout 3 days. So automatically we test the ruggedness, and also there are parameters which have been defined which are compatible with HPTLC. Now we'll move into the application. It has not got any defined one instrument with unlimited of possibilities, and that is why it gives you a lot of output. So if you have, for example, one department buys it, it can be a central instrumentation facility and everybody can utilize it widely for their research work in anything. When you start the reaction monitoring, you have two chemicals, they react and then form the third ingredient. So there you start. So HPTLC can be at that place. Then you move to the next step. It forms into a formulation which batch mixing is being done. So at that time, whether any impurity is being generated, whether it is the thoroughly mixed. So at that time you can test it. It is known as the content uniformity test. After that, the tablet is being done. The same content uniformity test can be also done over there. And then after that, you can, the packaging material, you can do accelerated stability studies, post-degradation studies, there HPTLC can done. And finally, after the packaging material is there, the packaging material studies can be also done. And like that, the entire process of manufacturing can be done in case of HPTLC. In pharmaceutics, this can be in food related aspect, where commercial foods are being done, or spices are done, or many things. You can do a lot, many things and many things. Like one people approached us for reverse engineering, where they wanted to see whether this final product has those initial compounds. That is possible. So these things are possible with HPTLC. We have a huge library of methods, and recently the method library is there in our instrument itself, that is in our software. Then you have an additional method library at our website of HPTLC Association, application notes at our principal space ChemMac, and you get a lot of books which are available. So like that, the huge database is there. Now we will not go thoroughly with all the applications. It will take a lot of time, but I will highlight you a few of them, and then we'll move into the video understanding of HPTLC. So this is how you see one experiment has been done of glimepiride. I'm sure you know about this drug. And here you see the separation being done on HPTLC with the validation parameters that have been studied. Similarly, apixaban, another tablet, biltefosine tablet, anti-malarial drug that is being studied. This is from a dissolution apparatus sample without any preparation, directly taken from the dissolution apparatus and applied straight away onto the plate surface, and you can analyze them. So your analysis is fast. Then you have degradation products, post degradation, simultaneous estimation of 3-4 compounds. Also FDC dosages can be done. Post degradation is there. Diclofenac from serum sample. So biological samples can be also studied with. Cleaning validation method. Cleaning validation is important after the reactor has finished the job, whether it is clean or not. HPTLC can help you out with that. Unknown impurities, where it is being used by a lot more pharma companies to identify what these impurities are. HPTLC can be a source of utility over there. Content uniformity, impurities, known and unknown. Now we move to the botanical applications. So identifying the proper fingerprint of the herb, proper herbs. Then you have kokum identity. Then you have tablets mixed with adulterant of synthetic material that can be detected. Then you have adulteration of other species, subspecies, curcuma longa adulterated with xanthorrhiza, and with a specific reagent, you get this blue color, and from that you can find out. Then you have steroids in herbal formulation. 4-molecule approach, simultaneous estimation in a herbal compound can be done with HPTLC. Now, food applications, fixed oils. Now, remember, if directly they don't relate to the pharmaceutical product, but at certain time they relate, like sugars, for example, they are used for syrups. You have oils which are used for topical applications in creams. So you can use these for alternative investigations. Aflatoxins, detection of sugars. Now a new approach, a new method from CAMAG has come, where 11 different sugar components are simultaneously being separated. Similarly, dyes also new methods have come. Okay, so just a minute, I'll just switch over my connection. Battery is low. Okay, then you have adulteration in oils, different types of oils, and here is an example where GC was being studied of the sample, but it would not be able to identify what components are there. But with HPTLC, you are easily able to identify what components are there because of the derivatizing reagents. And then you have oil adulterations, palmolive oil in normal oil detection that can be done. Preservatives, sample preservatives, you also use SMP and SPP, sodium methyl paraben and sodium propyl paraben as a preservatives in your many of the formulations, and that is it. We have an international symposium which is conducted, so anybody who is doing a good work and publish their paper, you get a good scholarship out of that, and ANCHROME helps you to publish if you are selected to present your paper with a certain amount of benefits to you. So you can utilize those. Last 19 and 20, we had winners from Indian scientists. Then unfortunately COVID was there, but now it has been resumed. So next year we might have the symposium. So all those, and finally this is our lab. I'll also show you the video which will explain you more. And with this, I will come to a conclusion of my presentation. Now we'll move on to the video session. So I'll just stop my presentation. Yeah. Okay, so I'll start sharing my screen once again. Okay, so now you can see my screen, and I'll just maximize this. And now let's see few components of our application. So first one is the ATS4. Let me just show you the ATS4 in glimpse. And okay.
Sample application and is suitable for high sample throughput in routine work. The instrument is software controlled and enables precise and reliable application of samples. The dialog box for instrument parameters offers user-friendly default settings. However, if you need to change predefined parameters, for example for quantitative analysis or for application of an aqueous solution, you can do this by just a few clicks. When working with default settings, it is only necessary to fill out the sequence table. The sequence table contains all the information which is needed to automatically run a series of samples. With its guided approach, the VisionCat's HPTLC software effectively leads you through the entire chromatographic process, step by step. Position the TLC HPTLC plate on the self-adjusting object support. After having loaded the ATS4, you can start the fully automatic sample application. VisionCat displays the current status of the sample application and the expected end time. An application sequence starts by rinsing the syringe. Next, the septum is punched and the needle lowered into the sample vial, thus allowing the syringe to be filled with the sample solution. The septum punch is retracted prior to the needle being removed to ensure the outside of the needle is wiped off by the tightly closing septum. The syringe moves to the programmed position of the plate and starts applying the desired sample volume. Several applications of the same sample are applied in sequence. Samples are applied as bands by default. Spots and rectangles can be applied as well. After the final application of the sample, the syringe is emptied into the waste and rinsed again, ready for the next application sequence. Once all samples are applied successfully, just open the cover and remove the applied HPTLC plate for chromatogram development, the next step of the HPTLC process.
So here you saw on the glass plate, this is the glass backing, and on that there was a silica. And this is the dimension of the plate are normally referred as length into breadth. So when we talk about that the plate size, breadth by length they talk about. So when I state the size of the plate is 20 by 10, so it indicates that 20 is the x-axis and y-axis is my 10. So the y-axis is on which I do the development and the x-axis is on which I will do the application. Now let me show you another video which will be of the developing device, and let me close it. And this will give you some clarity how the development actually happens.
Conditioning, chamber saturation, developing distance, and final drying can be preset and monitored using the VisionCat's HPTLC software. With its guided approach, VisionCat effectively leads you through the entire chromatographic process, step by step. The ADC2 will automatically develop the plate, leaving the analyst free to do other work. The ADC2 provides development under controlled conditions. Open the front door, load the applied plate in the plate holder and insert the plate into the automatic developing chamber. Pour in 10 milliliters of the developing solvent for plate development and 25 milliliters of developing solvent for chamber saturation into the solvent ports. The entire process is monitored. The plate is developed in a saturated chamber if using the default settings. The automatic developing chamber ADC2 and the optional humidity control create standardized conditions, obtaining reproducible results from plate to plate. Perform chromatography under different climatic conditions and compare chromatograms created in different labs of results obtained from different batches. Chromatography is started by lowering the plate into the developing solvent. During chromatography, the position of the solvent front is monitored. At the end, the developed plate is homogeneously dried in an airstream. All parameters are recorded in compliance with cGMP, cGLP as part of the analysis. After successful chromatogram development, just open the front door of the ADC2 and remove the developed plate for the next step of the HPTLC process.
This is the type of step where development we are doing, and now we are going to move to the next step. Yeah, just a minute. Let's start.
plates by employing a unique micro-droplet spraying technology. The standalone unit is available for two different plate formats, 20 by 20 and 20 by 10 centimeters. Select the small hood for HPTLC plates and the appropriate nozzle. To meet the diverging physico-chemical properties of the different reagents, 4 different color-coded spray nozzles are employed, and the user can select from 6 spraying levels. Recommended settings and recipes are provided. After positioning the plate, the hood is lowered and completely sealed to prevent spraying reagent from leaking to the outside. Fill the recommended volume of reagent into the nozzle. The spraying process is started by selecting the spraying level and activating the start button. The nozzle generates an extremely fine reagent mist, which evenly distributes in the hood and gradually condenses on the plate. Reagent remaining in the gas phase is automatically aspirated and collected in the wash bottle. When the spraying process is completed, push the lift button and remove the plate for further processing. Visual observation of the plate shows very high homogeneity. Reproducible and user-independent results are obtained from plate to plate. After completion of the spraying process, an easy cleaning procedure can be carried out. Simply rinse the nozzle with the appropriate cleaning solvent. And conveniently clean the hood with paper towels under.
Yeah, so this was about how the derivatization process happens, and now we'll see entire documentation. This is my colleague, Mr. Ramakant, who is explaining you. This is a very long video, but I'll only show you important parts which are related. This is actually shot at our lab. So this is the plate. You see, these are the plate boxes. If you see at the bottom, this is a typically aluminum back plate box. And along with that, at the beside, we have made a glass surface covered with aluminum foil. So we cut the plates on that. So these come in different dimensions, that is 10 by 10, 20 by 10 and 20 by 20 packages. And you have different numbers into it. So the aluminum sheet is there, and now he will show you how we have cut it. What we do is typically take this plate with the help of a steel marker. We are actually marking the plate. And once this marking is being done, we now cut across by sliding in. Okay, to cut the plate into 2 halves. And there you see the aluminum plate that is being cut into 2 halves, which is ready to accommodate 15 2 sets of sample into it. Now he's showing you by capillary spotting that over here we do, but normally we use the equipment, and this is how we place the plate. So he's pouring the mobile phase. Now he's typically showing you how a proper TLC step is being done. Generally, when we start with an R&D for quick, fast understanding, we do that and then switch over to the instrumentation. So here, after the application is being done, he has put the plate. The mobile phase is there, and I will just go a little bit back, show you how the plate is being put. So you can see he's having the gloves to avoid any fingerprint contamination. Slides over this, slides over and places the plate in one of the troughs. Now, twin trough, interestingly, if you see, the plate on the front end has a space and back end has a space. We normally put the plate on the back hand side, and the front end is used for saturation. Solvent from there that is being used with the filter paper. Now, in case of an ATS4, he'll do the same job as told, but through the software. So he has placed the plate and applied it. There is a rinsing bottle and waste bottle, and the application happens with the help of this applicator, as we saw earlier. Now, this is the developing device where we do the development. Again, this also module we have seen. It is being placed like this, and the plate is being placed inside. So this is how we load the plate inside the ADC2, and we attach it over there. And now close the surface of the door for the ADC2 and give the command through the software. The plate comes in and goes inside this chamber for the development, and we already have seen in ADC2 how it works. So humidity happens through this humidity control. You can see on the right hand side, there is a humidity chamber. I'll just go a few seconds back and show you how the humidity controls happens. So here it goes. You see, this is the chamber, and the air is being bubbled throughout to condition the surface of the plate. Now, after this has been done, the image is being taken in this visualizer. It has got 3 lamps, 254, 366, and a white lamp. These are universal, and the same set of lamps are used everywhere. There are selective filters onto the lamp to give you the selective light onto the plate. Many of the people don't have these filters, and that is why they see the plate in a different shade and color. But ours is optimally standardized. High quality filters are being used. So this is how we place the plate inside. This is the size of the plate, 20 by 10, as I was talking about. And you see over here that the sample is being applied and it has been developed over here. And after the development, he's adjusting this slot according to the plate size. It's a fixed focus camera. And that is why with a click of a button, you can find out the RF value and all. So after taking the image, we go into the next step, that is the scanner. This is the scanning device, TLC scanner. We place it with a magnetic strip over here and press the load button. When we press the load button, the plate goes inside and we close the door and give the command from the scanner. I have already explained you what are the different wavelengths from 190 to 900. Now this is another applicator, that is the Linomat 5. Here you have a syringe actually to load the sample. So we take the sample, we load it in this syringe, and then we place this syringe onto the Linomat 5 for applying it uniformly onto the plate surface. The only difference between ATS4 and Linomat 5 is the automation part. There the rinsing step happens automatically. Here we have to do the rinsing manually and load the samples each and every time. Now this is the AMD2 I was talking about. Here it is, and you get a vacuum pump which sucks out the solvent fumes after each and every development, and you load the plate on the top. There are different bottles, 5 different bottles of solvent which are there at the bottom, and you can unscrew them, load the plate inside this. This is the TLC MS interface, and there the gas connection comes in. I have a small video especially on TLC MS interface, and I'll brief you on that. So let us go quickly from this video. And this is the elution which is being shown. So there is this extraction lever on the right hand side. Solvent from the pump is coming onto the top. If you see, let me show you on the top. I'll go a few seconds back. And let me see. Okay. Anyways, we'll see a dedicated video of this. So here you have a laser pointer through which we accurately place the plate, and the spot which we want to elute is accurately marked in. These spots are first marked under UV with the set of a pencil, and after that we with the help of this pointer, we exactly locate the spot which has to be transferred. Then on the right hand side, that steel lever is there, which is the extraction lever. So by default, it is in bypass mode. It does not elute the sample. When you pull it in front, the elution head gets activated and the solvent is passed from the head and collects the analyte and sends it to the mass. So this is about the TLC-MS, and this is the immersion device I was talking about. For we have seen already the sprayer, the derivatizer. This is the immersion device. So here we load the plate in this way, and the glass dip tank you can see at the back of his hand that has the reagent, and simply he'll attach it onto that holder, and with the press of a button, this will go inside like this and come out, and the derivatization process is completed uniformly without any dragging or diffusion. This happens really rapidly fast, and this is the derivatizer. So this is about the hardware part. And now I'll just stop this video and we'll see another. I was talking about the TLC-MS video. So let us see a glimpse of that. So this is the TLC-MS interface. It is hyphenated with a mass spec of APSIX. And you have this lever, which is there for doing the extraction. So when I want it, I will just first flush it. There is a flushing button. If we press that, the head of the TLC-MS gets actually flushed in with fresh nitrogen, and then the head is being used for elution. This is the pump which is actually pumping the solvent into the TLC-MS interface. And this is how the connection, and on the right hand side, this is the lever which you are seeing actually. So when he'll pull this lever and the extraction is being done, it is sent into the mass. And after it is done, these are the spots which are being tested. That is caffeine sample. The parameters of the detector have to be known before you inject them. And that is what the tuning part comes in. And once that is done, when the extraction is being done, it is going into the mass and you get the spectra over here. So this is the background signal which you are getting. And the moment you get the injection, then you can see. So this is the background signal. And once the caffeine comes in, you will see a prominent peak of at 194. And that indicates that the, so we wait for a few seconds, 10 to 20 seconds, and then inject. So this is, you can see 197.94.8. That is because of the sodium addict that is being formed in negative and positive mode. You get this reading and you can see 195. This is the mass of the compound which is being seen. So this is the TLC-MS interface. We have more videos on our YouTube channel. So right now we are cutting this short, and hope this small presentation has given you an overall idea of how HPTLC is there, what we do with HPTLC, and how our analysis is being conducted with HPTLC. With this note, I am thankful for you all to be present and listening to this presentation patiently. Now I would like to open up this session for a few questions. We'll, as per the time permits, we can accommodate a few, and then rest of the questions you can send us at the mail at lab@ancrom.in, and that we'll be happy to answer them. I'm sorry. So let me go over here and check.
Sir, we have received some questions on chat box.
Yeah. See. Okay. Yes, I'm scrolling through. Yeah. One of the participants has asked an Ayurvedic injectable preparation which contains garlic tincture, nilgandi tincture, and Dashmuli Risht. Okay, but the injection you take I can. Yes, you can do their fingerprint and quantify them for the presence. You can correlate them, individual extracts. You know, the concept in herbal is always a BRM. That is botanical reference material or botanical reference mixture, and that extracts can be mixed in and one authentic sample can be prepared. The fingerprint of that will be serving as a quality control thing for each and every batch which passes out, of course, with certain deviations, accountability to the phytochemical variations. This can be passed that out of 10 spots, minimum 7 spots or 8 spots should be seen in the final formulation, and that decision can be taken autonomously. Apart from that, I'm not able to see any other question. Sir, can you guide me with any more questions which are there in the chat box? Or you can just unmute yourself and ask the question. I think so. This is a meeting, so anybody can unmute themselves. Yeah, I think so. Sunita madam has raised her hand. Can we have her unmuted, or you can unmute and ask your question, ma'am? Sunita ma'am, are you listening to my presentation? You can unmute yourself and ask the question.
Yeah, good afternoon, sir.
Good afternoon.
Sir, my question is the unit of injection, what is available for HPTLC, this system, sir?
Yeah, so in our syringes are in microliter. So 100 microliter syringe is with the Linomat 5, and you also get a 500 microliter syringe for preparative work. So you have to prepare normally like 1 mg per ml, or as I told you in the botanical extracts at that concentration. And using this needle, you can do those applications on HPTLC. So microliter, it is being converted automatically, and then application happens in microliter format. Once the application has been done, the solvent evaporates and only the microgram content on the plate enters, and anything related to it is being seen. So it is in microliters that the application happens. Is this giving your answer? If not, then you can just unmute and ask me.
No, sir, it's fine, sir. I got you.
Okay,
Sir.
thank you.
Sir.
Yeah, I think Mr. Gaurav, you have a question. You can just unmute yourself and ask me. Hello, Gaurav. Can you listen to me? Mr. Gaurav, sir, if you have a question, please ask it by just unmute yourself and ask me. I see your hand raised. Anybody else who has a question can raise their hands. This is a good approach. We can, you can unmute yourself and ask. I don't see any more hands. Okay, maybe that was by mistake. So anyways, do write to us at lab@anprom.in. Don't worry. We give answers and to all your questions, and your each valuable question will be answered from our side. So thank you so much for giving me this opportunity. And on behalf of this entire Ancrom team, I thank you for your valuable time. I have tried to give the capsulated knowledge of this entire HPTLC in a very short duration. If you visit in Mumbai and happen to come towards Mumbai, definitely visit us, and you can experience more and understand more for these sessions. We are also conducting few tabletop demos across the nation, and whenever it is in Orissa, you can come up over there, have an experience, and also get your queries clarified. So thank you so much for being with us, and all of you, all the best for your future. Thank you.
Thank you, sir, once again for your nice presentation. So it is very useful, and you have given clear information regarding your HPTLC and its applications. So I think our research scholars and faculty members will utilize this knowledge in their research field, sir. Thank you once again, sir.
Yeah, thank you, sir, for conducting this experiment, and special regards to the principal, Dr. Ravi Kumar, sir. Thank you so much again once, and also all the organizers who have taken the pen to conduct it, and so enthusiastic that even in Sundays, everybody is there. So it feels really nice. All the best to all of you, and whatever help required from our side, we'll always be there. So thank you so much.
Thank you so much, Dr. Saikat sir, for such a nice and illustrative session about HPTLC and its application. We truly appreciate your effort for sitting throughout the session and your lucid guidance. Especially the video demonstration is for sure going to help all of our faculty friends to refine ourselves about HPTLC and to gain insight about its different applications. Thank you once again, sir. May I now request our beloved principal, Dr. BVB Ravikumar sir, to felicitate our venerated resource person, Dr. Saikat Subir Kumar Malik sir, with an e-certificate of appreciation as a badge of our respect and deepest sense of gratitude from entire pharmacy faculty fraternity.
Thank you, Dr. Saikat, for accepting our invitation and enlightening all of us with the useful information on HPTLC. And I also thank Ancrom Laboratories for their help to Roland Institute of Pharmaceutical Sciences in various forms in the past also. So thank you, sir. And on behalf of the entire Roland Institute of Pharmaceutical Sciences and all the delegates who are present here, I would like to present this e-certificate of appreciation. Please accept it, sir. Kindly accept it.
Thank you so much, sir.
Thank you, sir.
Finally, I would like to take this opportunity on behalf of entire organizing committee, Roland Institute of Pharmaceutical Sciences, to place on record our hearty thanks to Honorable Vice Chancellor Sir, Director Curriculum Sir, BPUT, Dean Pharmacy, directors and principals of all BPUT affiliated colleges, above all, resource person of today's event, Dr. Saikat Sir, for sparing his valuable time for us. We are really enlightened with their knowledge and their presence. We're thankful to our Honorable Management Committee RIPS for their constant support and motivation in all dimensions to carry out such programs. I would like to thank our principal, Professor Dr. B.V. Ravikumar Sir, for his enthusiastic support throughout this program. A special thanks to the program coordinator of this event, Dr. Viswamohan Sahu Sir, for all the timely arrangement and his precise efforts for the smooth conductance of this event. My special gratitude to all the members of the organizing committee, teaching and non-teaching staff member, all the faculty delegates friends for their unflinching support and coordination. With these warm words and kind messages from our eminent personnel, we now officially conclude today's online faculty development program on HPTLC and its application. Thank you very much all the faculty delegate friends for sparing your valuable time and your kind cooperation. Thank you all once again.